Good evening, everyone. I'm Susan Fernbach, and along with Dr. Deborah Murray, we are the co-directors for the Office of Community Engagement and Diversity for the Department of Molecular and Human Genetics at Baylor College of Medicine. We welcome you to this Evenings with Genetics seminar tonight, honoring Black History Month. This is the second of the webinars on race and genetics, perspectives of precision medicine. And we had to reschedule due to the prolonged February ice storm, which shut down power. And we thank the speakers for the accommodations they made to be with us tonight. This is really the 15th anniversary of the Evenings with Genetics webinar series. It's a free series for the public, and it began as in-person monthly seminars at the Children's Museum of Houston. And the goals have always been to provide current genetic and genomic information in a plain language, concise and clear manner, and to offer support and resources for those impacted by genetic disorders, as well as to foster collaborations. And we hope that that happens tonight. Due to the ongoing pandemic, the series has moved completely virtual, so we welcome all of you from whatever part of Texas or the US you're joining us from. We also thank Dr. Brendan Lee, the chair of the Department of Molecular and Human Genetics, for his support and encouragement for this series, as well as the many faculty and family speakers who've shared their expertise over the years. The series is planned by the Evenings with Genetics Committee, and a special thanks to Dr. Neil Hanchard for his role in planning the February webinars. Due to the ongoing social issues in our country, we felt the strong need to address the old concepts of genetics and race to help us help more people in all of our communities. These two webinars are sponsored by the Department of Molecular and Human Genetics, as well as the Baylor College of Medicine Office of Diversity, Equity, and Inclusion, the Human Genome Sequencing Center, and Baylor Genetics Labs. We are so pleased to introduce our three speakers tonight, Dr. Charmita Hughes Halbert, Dr. Rick Kittles, and Ms. Hoxie Jones. During the webinar, you can enter questions in the Q&A box at the bottom of your screen, and we will get to as many questions as possible. So let me introduce our first speaker, Dr. Charmita Hughes Halbert. She is the Associate Dean for Assessment, Evaluation, and Quality Improvement, and professor in the Department of Psychiatry and Behavioral Sciences at the Medical University of South Carolina. Dr. Hughes Halbert earned her Ph.D. from Howard University with a focus on personality psychology after completing a fellowship in cancer prevention and control at the Lombardi Comprehensive Cancer Center at Georgetown University. She was then recruited to the University of Pennsylvania in the Department of Psychiatry and later promoted to associate professor, becoming the first African-American woman to be promoted with tenure in the department. In 2012, Dr. Hughes Halbert was recruited to the Medical University of South Carolina as professor in the Department of Psychiatry and Behavioral Sciences and the AT&T Distinguished Endowed Chair for Cancer Equity at the Hollings Cancer Center. Dr. Hughes Halbert was elected to the National Academy of Medicine in 2017, becoming the first investigator and woman in South Carolina to earn this distinction. And it's wonderful to tell you this on International Women's Day. Her research has been in genetic counseling and testing, and she has provided a model of culturally tailored strategies for cancer prevention and control among racial minorities, and she continues to advance the science of precision medicine in minority populations. So with a warm welcome, I turn it over to you, Dr. Hughes Halbert.
Well, good evening, and just wanted to start by thanking the conference and webinar organizers for inviting me to participate in this important webinar. I'm really excited to be here because we all are sort of, at this moment of time in our individual and collective social histories, we're just, it's it's great to sort of have a space and a venue to talk about issues related to race and genetics, and have an opportunity to think about how we need to reimagine those concepts as we move forward. So I think what I wanted to do is just start with setting a stage for how we think about race and how the potential for using genomic and genetic information as one component or one strategy for promoting health equity through individualized approaches for detecting diseases earlier, for providing prevention and treatment. And really, when we think about this, or when I think about this, the ultimate goal is to ensure that individuals have the right care at the right time. I think there's, we are, as you were talking and sort of going over my background, my professional background, it really made me think about how much the field has matured. And what I'm showing you now is a relatively recent conceptual model of minority health and health disparities that I think has influenced all of our thinking of racial disparities and minority health and health disparities, and thinking that we really have to think of determinants with a multi-level perspective. So as trained as a personality psychologist and doing behavioral science research, I tend to focus on sort of this individual level as it relates to health behaviors and coping strategies. But there's also important to think about behavioral strategies, behavioral determinants, as they have influence at individual, interpersonal, and community levels. So this is really, I think, an exciting time where we're all trying to work together as part of transdisciplinary teams to have the best understanding about the complex ways that determinants at multiple levels of influence impact disease risk and health outcomes. So I was really excited when the Precision Medicine Initiative was announced, now I think in 2016, and it's evolved as all things do into the All of Us research program. I was excited by the Precision Medicine Initiative because I saw an opportunity for someone like me with my background and expertise in behavioral science be part of groundbreaking efforts to develop prevention and treatment strategies that are based on individual differences in genes, environment, and lifestyle. And I think, we know that health behaviors are complex. We know that disease outcomes and disease risk are complex. And so the Precision Medicine Initiative, to me, really set the stage for giving us an opportunity to work across multiple domains and multiple levels of influence. One of the things that I think has emerged as a priority as a result of the Precision Medicine Initiative is a new focus on data science. And what I'm showing you here is just this kind of word cloud that shows all of the different permutations and ways in which data science is important in the healthcare setting. And so our ability to take data, understand it, process it, and derive some value from it, and then ultimately communicate it are some of the critical elements in precision medicine. But data science, I think, and the ability to communicate data has been an important issue in minority health and health disparities, because when I think about it, I think about being able to take information about a genetic risk factor and communicate that, translated into an effective communication strategy that will be beneficial to different groups. So data science, I think, the field has certainly grown, but it's one that I think started with some very important foundations in risk communication and sort of the how we think about communicating genetic risk information. We're now at a point in the field where we now have a focus more on social determinants. And what I'm showing you now is just different ways in which social determinants are conceptualized and can be measured. to me, I think an important point in development in healthcare and healthcare delivery and clinical research and also in genetic research is really being able to understand someone's social environment or social background and to understand that how that influences the delivery of healthcare, decisions about treatment and management at an individual patient level, but also as it relates to population health. An important point shown here is that we really have to work towards obtaining social determinants data from patients, documenting that information in a way that's usable for healthcare systems and providers and patients, and using that information to address unmet social needs. As I've been thinking about social determinants of health, and the slide that I just showed sort of talks about, it illustrates housing, income, poverty, access to healthcare. What's been interesting to me is that race is not included in that illustration. And I think one of the reasons why it's not included is that there have been some efforts to move away from thinking about and talking about racial differences because it's an emotionally, socially charged word and descriptor, particularly as it's sort of linked with genetics. I take the view that race is an important social determinant that structures and influences how we experience the world and how we experience healthcare. And what I wanted to do is just share with you a couple of examples of the ways in which different types of social determinants are being addressed in my own research, and certainly influenced by others in the field. So one example that I'm showing you now, and I think this has important implications for genetic research, is that a primary focus of our efforts to understand racial differences in any disease risk or outcome has been the enrollment of racial and ethnic minorities in cancer clinical trials. And so I'm showing you a report from the Cancer Letter that was published, I think the date is in 2020, June 26, 2020. And this was an important publication and report because it demonstrated that although there still is a way to go in terms of proving equitable access to clinical trials, that there have been improvements in terms of the representation of diverse populations in cancer clinical trials as a result of programs such as the NCI Community Oncology Research Program. There's been a lot of focus on understanding race as a determinant of recruitment and participation in cancer genetics research in particular. And what I'm showing you now is an example from work that was done when I was at the University of Pennsylvania and led by my postdoctoral fellow at the time, Jasmine McDonald, where we were really wanting to understand what makes it easy and difficult for individuals to decide to participate in cancer genetics research. And then lastly, how do those participation barriers and facilitators interact with the attributes of the study to influence willingness to participate in cancer genetics research? So we conducted, I was really proud of this work for a number of reasons, one of which is that we sort of took a perspective of understanding the actual experiences of individuals who have participated in research, who declined participation in research, using a mixed methods approach and developed, I think, some really important measures as a result. Our final work from this study, I think, really resonated with me with the sort of the overarching framework of the Precision Medicine Initiative. And one of the vignettes that we presented to a national sample of African Americans was a study that had different attributes, which are shown here in the upper left-hand side of the screen. And so we asked individuals to tell us how likely is it that they would participate in a study that was sponsored by the government, that involved answering a questionnaire. Their data would be used for current and future studies, and participants would not receive results. So as we developed these vignettes, an important consideration for us was to integrate some of the ethical, legal, and social concerns that had been identified as potential barriers among participants in our qualitative interviews and in our quantitative survey. You can see that only about 31% of participants said that they would be willing to participate in this type of research. And it really illustrates, I think, an ongoing persistent issue in the field of genetic sciences, in that there continues to be an underrepresentation of African Americans and other racial and ethnic minority groups in the cohorts that are being used to make new discoveries about the contribution of genetic factors to disease risk and outcomes. So this study provides some insight about the things that would need to be addressed as part of enhancing participation decisions. The other social determinant that has really raised in terms of its importance is geographic issues. There's sort of, catchy terms called the community anomics, the Z factor. I'm forgetting what the name of that like catchy, what that sort of the catchy name for zip code. But there's been a huge focus previous to sort of this sort of reemergence of understanding where and how individuals from diverse backgrounds live, work, and play. And a focus that our group sort of, we addressed it through using a community-based participatory research framework. We did several different studies, which were all focused on actively and meaningfully engaging diverse community stakeholders in the process of, one, defining the research topic of priority, and then working with collaboratively as part of an academic community partnership to combine that knowledge and action for social change. Our goal was to improve, and in this study was focused on individual health behaviors, but our subsequent outcomes are really sort of had a broader and I think more aspirational goal to change the actual community environment. This is important because as we think about, where we are now, there's sort of like this complex interaction between race, where individuals live, or environmental factors, and genetics, and they all sort of work together. And I know Rick is going to talk about some of that as in part of his talk. So this is who we are and what we did. It was MUSC once I moved here, and then the Health Promotion Council of Southeastern Pennsylvania, the University of Pennsylvania, and the National Black Leadership Initiative that formed this academic community partnership called the Triumphant Living Collaborative. We did a comparative effectiveness trial that was based on the priorities of our community residents. You can see the outline and flow of the intervention sessions. Our primary goal was to promote and increase the likelihood that individuals would engage in health promotion and disease prevention behaviors, particularly increased fruit and vegetable intake and physical activity, by sort of motivating them through improving their knowledge about risk factors, overlap and shared risk factors for cancer and cardiovascular disease. We found that, and this shows our sample, 530, individuals who lived in the Philadelphia metropolitan area. I'll draw your attention to one thing that will sort of be a theme as we move forward, is that 43% of our sample were African American men, which is, really impressive, I think, for that time, and even for today, because there tends to be an underrepresentation of African American men in particular in health promotion and disease prevention research, as well as genetic studies. So I mentioned that neighborhood was really important. And as we think about, I think some of the new initiatives that are coming out that have a focus on addressing and mitigating structural racism, for example, some of our work has illustrated that individuals don't perceive like a huge amount of efficacy in their ability or their confidence in their ability to change where they live and how they live. And I think that's really important because it shows that, as we sort of think about how we can change an important social determinant, that geography, that there's likely to be a lot of work that needs to be done to address sort of enhanced levels of confidence in making those changes. So we found that we had high levels of participation in the intervention and completion of the intervention. I think that was because we actively worked with community representatives from community-based organizations, as well as community residents to help us think about and implement strategies that would be effective in terms of enrolling participants into our research. And the key finding was that there was a significant increase in the proportion of residents who met the recommended guidelines for physical activity. But when we controlled for variables associated with retention in the study, it was really whether or not they completed the intervention and their education level that were associated with short-term changes in physical activity. An important issue with respect to intervention development is that, and particularly from a community-based participatory research framework, is whether or not it addressed concerns. You can see here that participants reported that our interventions did address their concerns related to physical activity, fruit and vegetable, and fruit and vegetable intake. So here we are now, and so this is, by this point it's 2015, 2016. And, I've moved to the Medical University of South Carolina. And one of the things that became really apparent to us was that one, you can enroll and sort of address the needs of African American men with a particular focus and having that as a priority. And so again, sort of thinking about race and gender as a particular focal area. The other thing that became apparent to us as an investigative team was that there is this, unique unique experience that African American men have. And it led us to be really focused on developing approaches and strategies that would address some of the primary determinants of morbidity and mortality. We started this work within the context of prostate cancer. And one of the things that we found was that perceptions of stress were important to how men reacted to their initial diagnosis and their emotional and physical well-being following their diagnosis and treatment. This again sort of reiterated the point to us about psychosocial stress and its relationship and influencing in cancer outcomes. And we operated from the perspective of, if you're experiencing a lot of stress in your just general life, it's not likely to get better following a cancer diagnosis. And as a result of that, we sort of focused our work now on understanding the ways in which psychosocial stress and stress, in the environment impacts biological functioning. And so we're doing that by using a measure of allostatic load, which reflects the extent to which social and psychological stressors impact biological functioning. Our primary question for our Precision Medicine Center is, again, how does psychosocial stress influence cellular stress? And these, have complex sort of mechanisms involved. Certainly the stress response is not just about how you perceive it to be, whether or not you perceive it to be stressful, but the extent to which it activates the HPA axis, which has all different types of mechanisms that are involved, some of which involve genetic determinants. So as we begin thinking about our work and sort of how we can have an impact, one is sort of, again, focusing on African American men as a particular focus of our center. We really wanted to develop tools and strategies, sort of remembering that one of the primary drivers of precision medicine is data. How do you integrate big data? How do you use big data as part of developing population health strategies on into individual levels of care? This shows our transdisciplinary sort of framework for data science. And it really has been exciting for me to be part of this team because it brings into focus expertise in behavioral science to help bring into the discussion, and research framework, determinants of minority health and health disparities, brings in informaticians who bring expertise in developing and applying natural language processing tools as informatics strategies, and then geospatial issues and bioethical expertise to understand, well, what should we do and what should we not do as it relates to representing geography. This shows an example of how we've worked together as part of our team to sort of link behavioral science and machine learning and informatics expertise. One example is sort of, behavioral scientists help us to understand, well, what are the most important social determinants that we need to identify and extract? Then our machine learning and informaticians help us to develop the tools to be able to extract it. And I think as we think about race and genetics, it's important to begin applying some of these tools to the existing data sets that exist to help us understand better and leverage the data that we're using. So this is an example of the NLP pipeline that has been developed as part of our team. And I wanted to share with you one example of a lexicon that was created to identify social isolation. And it's really kind of telling because the tools that are developed and implemented, reflect what is either discussed in the clinical scenario or what providers believe was important to document. And I think that's really important because we have to think about what types of data and information we collect as part of initiatives that are sort of focused on linking race with genetics. And then in the last example that I wanted to share with you is sort of our data science approach for allostatic load, with just a reminder that allostatic load reflects physiological responses to stress. And there are several different ways in which you can determine allostatic load using vital statistics stored in the electronic health record, vital statistics that are obtained from laboratory assessments, and then stressors that are self-reported. We've been working to understand the nature and distribution of allostatic load in our different samples. And one that I want to leave you with, I know I'm running out of time. One that I want to leave you with is that as we think about different models for allostatic load, so this bar chart shows the outcome of a model that's really based on clinical determinants. So, and these are clinical variables that are important to prostate cancer risk. So African-American men have higher allostatic load. There's also a positive association between age and allostatic load. When we control into the model with this social determinants model, these variables all sort of become non-significant. It's really negative life experiences that contribute to that have a significant association with allostatic load. Again, sort of raising the point of, if we think about sort of race, a clinical indicator or a variable that's influenced by genomic processes, it then becomes a social characteristics that are most important, which is shown here in this next slide. And I'm gonna click through some of these very quickly. Well, I said that was the last example, but the very last example I want to give is shown here, where, as I've been thinking about behavioral science research, for those of us who sort of were trained in a traditional psychology department, we think about behaviors in terms of self-reported physical activity, self-reported dietary behaviors. Observe, then there are all different kinds of ways to measure that with more or less precision and accuracy. I'm really excited now by this field called basic behavioral science research, which is really focused on linking sort of biological mechanisms with health behaviors. And I think there's a tremendous opportunity to understand the ways in which genetic variables influence behavioral responses or influence the sort of the the precursors to the behavioral response, one of which that we're examining in our research is the glucocorticoid receptor gene and genotypes within that receptor gene and how that influences stress responses, and then ultimately how that influences glucocorticoid resistance. And then lastly, how that leads to cancer risk behaviors. So I will end my talk there, just again by thanking the conference organizers for the opportunity to participate in the session, and I look forward to the discussion. Thank you.
Thank you very much, Dr. Hughes Halbert. And we will have questions at the end, but I'd like to introduce our second speaker now, Dr. Rick Kittles. Dr. Kittles is a professor and founding director of the Division of Health Equities within the Department of Population Sciences at the City of Hope in California. He's also the Associate Director of Health Equities at the City of Hope Comprehensive Cancer Center. Dr. Kittles is well known for his research of prostate cancer and health disparities among African Americans, and his research has focused on understanding the complex issues surrounding race, genetic ancestry, and health disparities. Dr. Kittles received a Ph.D. in biological sciences from George Washington University. His first faculty appointment was at Howard University, and he helped establish the National Human Genome Center at Howard University. Over the past 20 years, he's been at the forefront of the development of genetic markers for ancestry and how genetic ancestry can be used in genetic studies on disease risk and outcomes. In 2010, Dr. Kittles was named in Ebony Magazine's The Ebony Power 100. And in the March of 2012, he presented the keynote address to the UN General Assembly on the International Day of Remembrance of Victims of Slavery and the Transatlantic Slave Trade. Recently, Dr. Kittles was named by the Huffington Post as one of the 50 iconic Black trailblazers who represent every state in America. So welcome, Dr. Kittles.
Thank you. Thank you very much. I'm excited to be here, and this is a very important topic that's near and dear to me. And so we should have some lively discussions towards the end, I'm sure. So I'm just going to talk briefly about race and genetics and the role that race and genetics has played in biomedical research, but also the impact that it's now having as we roll out precision medicine or personalized medicine. When we think about race, historically it's been a crude proxy for some shared biology or shared environment. And it's historically been a crude proxy for some risk factor. Let's say, for instance, one goes through the literature and sees that race is a predictor of stroke, race is a predictor of end-stage renal disease, race is a predictor of prostate cancer. But that doesn't say much about the biology, if biology is contributing or if it's the environment that's contributing. And so we're moving away from race in this setting, where we're actually now measuring the individual's ancestry, their genotype, their genetic background. And we're also measuring in much better ways the environment, be it diet, lifestyle, exposures. You heard a beautiful representation of that from Dr. Hughes Halbert. Where we're actually looking at this intersection of race and the physical and social environment and how that impacts on how individuals see themselves and the like. So when we measure those separate entities, it allows us to assess if that complex disease is due to the biology, due to the environment, or due to, in many cases, which it is, the interaction of the two. But you can't get that if you just look at race. And that's why this is so important when one talks about genetic ancestry and measuring ancestry and ancestral background. Now, I'm not saying that we throw away the baby with the bathwater and not use race. Race does have some saliency in society. It's a social cultural concept, but it doesn't provide useful insight in terms of biology. So when we talk about ancestry, and in particular disparities, one of the things you see is that most communities of color, be they African American or Hispanics in the US, are very biologically diverse. And when we talk about the African American population, a lot of that diversity is because of the antiquity of the African gene pool, diverse West and Central African ancestry for most African Americans who are here today, who are descendants of enslaved Africans. And then there's gene flow that came into the African American population from non-Africans. And a lot of that historically, when we look at the history, most of that has been male centered or male directed gene flow from white men into the African American population. The pattern of variation differs geographically also. So when I was talking about this antiquity of the African, West African gene pool, this is where the bulk of African Americans gene pool, 95% of the African gene pool comes from West and Central Africa, from Northern Senegal to Southern Angola, 5% from East Africa, Mozambique and Madagascar. But you got to understand that there's enormous biological diversity in this area of West and Central Africa. And there's also a lot of cultural diversity too. It's not like there's just monolithic African, just, you know, historically we've used the Yoruba samples in the HapMap cohort to represent all of Africa when there's enormous variation. And we're doing a lot better now in terms of sampling Africa, but I'll get into that shortly. For some reason, this isn't turning like it's supposed to. The genetic ancestry helps uncover the genetic structure at the individual and at the population level. And this is where things get really, really neat, because one can then assess and explore how this shared ancestry differs and how it's similar across individuals within certain populations. But what's fascinating is that we see this mosaic of chromosomes, where admixed populations, where we have a punitive population, ancestral population, and another ancestral population. Let's say this is European, this is West African. The initial mixing, 50% of the genome coming from either population, but then over time we have this reshuffling, this restructuring, this recombination that occurs across those chromosomes. And they leave with it this signature of a mosaic ancestry. If one were to color the different ancestral proportions, let's say blue and red. Some individuals, depending on when that European ancestor is in the family, may have bigger stretches across these chromosomes of European ancestry. And this is important to understand. So we use markers called ancestry informative markers to, historically, we've been using that to estimate ancestry, but now with the current knowledge of SNPs and the allele frequencies across the world, one can assess using GWAS data or sequence whole exome or whole genome sequence data with pretty good accuracy, some level of ancestry for the individuals. This is a plot showing 4 populations, the spread of ancestry across these 4 populations. The black here represent Nigerian West Africans, the red are African Americans from Chicago, the blue are whites from Chicago, and the green are CEF French samples. And you'll notice here that there's a broad spread of variation here. But it's a continuous spread. There's no clear disconnect. There is no, this is black, this is white. These individuals self-reported across the board, from African all the way to European. But you'll notice that there is a lot of overlap amongst these samples. For instance, some of these red here are just as European or more European on this genetic spectrum as some Europeans. And so how one defines themselves is really important. We've always said self-identified race and ethnicity is an important factor, because it carries with it some information about how those individuals are socialized, how they see themselves, and it may not necessarily correlate with their genetic background. And this is important, especially as we move forward into precision medicine. So African Americans aren't the only ones that are very heterogeneous. I love to show this. This is from a colleague of mine, Carlos Esteban Bashard, who did a study looking at Puerto Ricans and Mexicans who with asthma. And here you see the distribution of ancestry, individuals along the x-axis, and then the proportion of ancestry on the y-axis. And you'll notice that the proportion of African ancestry is much broader and much more significant among the Puerto Rican samples than the Mexican samples. And this says something also, as I mentioned before, about the heterogeneity within the Hispanic and Latino population. You know, we grouped them together because the federal government says they speak the same language, Spanish, they should all be grouped together, but genetically very, very diverse, very different ancestral proportions that could impact genetic disease studies, but then also precision medicine approaches. When one, now, as I mentioned before, we can go marker by marker across these chromosomes and measure ancestry from the major geographic groups. So for instance, here is a stretch on chromosome 5 of individuals from West Africa, Europe, and then here are African Americans. And you'll notice that the West Africans and the Europeans, pretty homogeneous there in terms of blue and red, but the mosaic of chromosomes here for African Americans is what's really interesting because it's reflective of the ancestry of those individuals, of the family history of these individuals, some having much greater proportion of European ancestry or more recent European ancestry, while others may have less or more distant of European ancestry. So we can do that now using these markers that have been identified across the chromosomes. So this, I like to show this. This is my chromosome painting. This is my genome, Rick Kittles, not 100% West African like I thought, but about 90% West African, 10% European ancestry. And a lot of that European ancestry is because of my Y chromosome also is of European ancestry. But when you look across these chromosomes for me, you see the burgundy, which is African, and then the blue representing the shades of blue representing Europe. And you'll notice that for instance, almost all of chromosome 2, except for this tip here, is of West African ancestry. So I have African alleles across the chromosome for genes that are on that chromosome. For instance, chromosome 6, there's no European. But then you have, for instance, with me here, almost a quarter of chromosome 10, or maybe even more than that, is of European ancestry. I like to bring folks' attention to chromosome 15. I have, for the entire stretch of chromosome 15, African alleles, which are important for these particular genes here on chromosome 15. And I'm sure most people are wondering, what is he talking about? Well, the biggest predictor, the two biggest predictors of skin color are on chromosome 15. And while I have 10% European ancestry, you may not see that reflected when you see me walking down the street. You say that's an African American, he's of African descent. And that's because all of my chromosome 15, in particular across those two genes that predict skin color, I have West African alleles for those particular genes. This is important because what we can do is we can leverage local ancestry to map genes for diseases that disproportionately impact these, that are disproportionately more prevalent or higher mortality between the two parental populations. And we've done that for prostate cancer. Many have done it for other diseases too. But it's a good way to leverage where we find this enrichment of these disease alleles or drug response markers to leverage ancestry to find those and to see what the important variables are. So when we look also across the United States, we find that the pattern of genetic variation differs and it varies geographically and socially. And this goes to this issue of local experiences and local histories. They do matter. When we look across the United States at the proportion of European ancestry in African American communities, we find it varies. And in the rural South, it's a lot lower than in the urban North, where it's more cosmopolitan, a lot more mixing. But I'd like to bring this attention because we were in awe at Dr. Hughes Halbert's talk earlier. And some of it was representing South Carolina. when we look at South Carolina, in particular among the Gullah, it's about 3.5% European ancestry, very homogeneous in terms of West African ancestry, not a lot of European ancestry. In Charleston, about 10%, and then we go to Columbia, South Carolina, it's about 18%. So all throughout this area, much of what is considered low country, it varies considerably, the proportion of European ancestry in the African American population. And I find that to be quite interesting. And even within the rural communities, different local histories matter. Columbia is the capital, it's been the state capital, major highways, major university, and the behavior of folks in Columbia is quite different than the Gullah Sea Islands off the coast. I'd like to bring attention, 3.5% on the Gullah, 35% European ancestry out in Seattle, Washington, vastly different social norms, local experiences, and and different behaviors of Blacks and whites. And because of that, did my screen stop? I'm sorry. Yep, so the 10% higher than what we find in the Gullah Sea Islands. And this is reflective of what we see throughout the country, where we see where local history does play a role in the mate selection and the distribution of these particular alleles in the African American population. So when I talk about race, I'm talking about the social construct of race. I don't use it in a biological sense. When I want to talk about biology, I talk about ancestry and genetics, but there is this interaction between the two. And a lot of that is mediated based on skin color. So how one views African Americans and Latinos, a lot of that is this interaction, the biology and the social is mediated through skin color. So when we talk about precision medicine, this is where things get interesting. So, we historically for a long time have studied white men. And this is why we're in the point, we're in the space that we are in now, where precision medicine isn't actually benefiting all communities equally. Precision medicine is this rapidly emerging field, providing insight across the board in new drugs, biology, pharmacogenetics. But the bottom line in this is that what we're trying to do is intervene earlier in the disease process. So if we look at this red line for the disease process, the typical current intervention is late in the game. And when you're late, outcomes are poor. You want to intervene earlier. And that's why we're using the tools, the precision medicine tools and techniques to intervene earlier, whether it's assessing risk all the way to prediction and prognosis. But the issue is most of the information that we have is based on populations of European ancestry. And so because of that, and because of the fact that the bulk of the data is on whites, it's very expensive to for many of these tests. It may actually increase health disparities. These tests are currently available and folks are taking a very active role in clinical decision making. However, as I mentioned before, it does have, there is this, more folks are becoming much more aware that actually precision medicine may increase disparities if it's not equitable in terms of benefit. I just wanted to give an example of a precision medicine sort of approach that we did and showed that, this is very important for diverse communities. So naltrexone, which is a drug that has been used historically for treating alcohol abuse, and then it became useful in smoking cessation. However, historically, it's been shown that whites are more, there's more efficacy of the drug in whites than it is for African Americans. And so I was able, when I was in Chicago, to get a hold of a data set from a clinical trial. It was a smoking cessation trial among self-reported whites and Blacks. And I saw a very similar, what was found in the literature. I found it in the clinical trial where the African Americans just were not responding to this drug like the whites were. And so this is just a survival curve showing the difference in response between the treated and the untreated groups. But there was no separation here of the line that was significant here between the treated and untreated among the African Americans. So what we did in this trial was we went in and said, why don't we, since there appears to be this biological difference, let's see if it's really biology, or maybe it could be, some demographic, some social behavioral component. We went in and said, okay, well, for the African Americans, we're going to type ancestry, measure ancestry, and then stratify the group based on low West African ancestry and high West African ancestry. And when we looked at the whole sample, the pooled sample of African Americans, there was no difference here for one of the outcomes, which was a 4-week quit rate. No difference between the treated group, the naltrexone group, and the placebo group. But when we looked at the African Americans that had high European ancestry, or what we call low West African ancestry, we found individuals who benefited from this particular drug compared to the group that had high West African ancestry. So we said, oh, well, maybe there is some genetic marker that's actually contributing to this difference that we see. So ancestry in this situation was used as a biomarker for assessing if these individuals were going to respond or not. But the real question was, what is it about the genetics of these individuals that made them more responsive? So we then went in and sequenced one of the big genes that's involved in the response for naltrexone, which is the mu opioid receptor, and found that there were significant variants in the promoter that were restricted in terms of allele frequencies to West Africa. And that contributed to, these are functional variants that actually contribute to the lack of response in the African American, the high West African cohort of African Americans. These individuals genetically were more similar to Europeans and actually were able to respond differently because of that similarity. Now, we just had a cutoff of the median cutoff of ancestry. And so while this is important to understand, it's not, there is no strong clinical utility, because we're assuming that ancestry here is acting as a proxy for those genetic differences that were important for the drug response. And we have to do a lot more work. The bottom line is we have to do a lot more work in order to really assess the role that ancestry is going to play in the clinic. What it does do, though, is it allows us to dig deeper and figure out what those differences are within those groups, to figure out what the important functional variants are. And so I'll end there, and I'd love to take questions when this Q&A comes up.
Thanks very much, Dr. Kittles. This is great. So we have a third speaker now, Ms. J. Hoxie Jones. And Ms. Jones is a sickle cell elder advocating for sickle cell disease for more than 40 years. Ms. Jones has served on many committees throughout her life, both locally and nationally, and has shared her journey with healthcare providers and families. She was very helpful during this most recent February ice storm in providing lots of resources, and I am very thankful for that, Hoxie. She enjoys a leisurely retired life and lives in Houston, but I can attest to her being a very busy woman. So we really look forward to hearing from you, Ms. Jones, and your perspective as an individual who's been in the healthcare system and receiving care within it. So I'll let you take it away.
Thank you, Ms. Fernbach, for inviting me to be the last panelist for today's Evenings with Genetics. Over the years, I have learned so much from these forums, and tonight is no different. Many times the information is like, but I still glean some nuggets from it. When I read the introduction on the webpage about tonight's topic, do racial categories obscure our genetic similarities and differences? It made me pause for a moment and say, really? As a 67-year-old woman living with sickle cell anemia, I have survived this question throughout periods of oppression to my body from this disease, followed by intervals of freedom, allowing me to operate unimpeded in daily space. There is a lot of courage that goes into sustaining a normal life when you can't anticipate the interruptions by the specter of oxygen-deprived red blood cells turning from their pliable donut shape into rigid, sticky rods that characterize the unimaginable pain and damage. And this pain is usually what places me on the opposite side of the healthcare corporation, most notably hospital emergency departments. It gets so bad there is no other recourse but to go there for relief. The trepidation stems from not knowing who is going to show up, someone who is caring or someone who is waiting for their shift to be over. Because sickle cell disease, or SCD, is more common among people of African, Caribbean, Middle Eastern, Eastern Mediterranean, and Asian descent, the burden of illness is greater than perhaps it would be if dominated by people of the global minority or white people. As a Black woman, there is no way to cover this aspect of my being up. That is how the world sees me. No matter what venue I happen to be in, superimpose the opioid crisis and resulting deaths with ill-conceived policies that disproportionately affect people of color, and we have the perfect storm of bias exhibited by those we seek compassionate care from. There are things, however, that have improved since SCD was described in 1910. Hydroxyurea to improve the course of the disease in adults, along with stem cell transplant and gene therapy, tools that may lead to a better quality of life and provide hope for those of us living with this debilitating affliction, are available because of research. We all know the history of clinical trials, the negative impact to people with Black skin in America, and our reluctance to participate. But these therapies are a result of clinical trials, so they are important to advancing the science from bench to community. A better job of informed consent may increase a person's understanding of the benefits, risk, and possible health outcomes. I recently read about a new therapy that may reduce the use of pain medicine. According to data from a phase 2 clinical trial on vascular electrical stimulation therapy, or VEST, a form of local electrical stimulation is applied to the skin, lowering the duration of the painful vaso-occlusive crises in patients with SCD. This made my eyes light up with excitement. What if there is something besides narcotics? On the other hand, on February 16th, there was an announcement regarding the temporary suspension of Bluebird Bio clinical trials of letin-globin gene therapy for sickle cell disease, and the pause of all commercial use of Bluebird Bio European gene therapy. Then Aruvant disclosed on February 23rd that they paused their gene therapy trial for the gene transfer for patients with sickle cell disease as well. Communiques like these cause skepticism in patients and families, but I know from personally participating in clinical trials, the only way we're going to beat sickle cell is by discovering what works and what doesn't. I can remember decades when there was a vacuum of clinical trials in this disease. Sickle cell is a chronic, progressive disease existing from birth to grave. I am privileged to live in Houston and be a patient of UT Physicians Comprehensive Sickle Cell Center, an oasis of experts who take time to not only care for you, but educate you along the way. It makes a difference when you have people who see, value, and understand you. The critical nexus of insurance and qualified healthcare providers stands to be impacted by our DNA. How long after whites get precision medicine will we be afforded the opportunity? Are we as Black Americans and people of color who represent the global majority worth it? Time will tell. With age, there are insidious complications from the daily onslaught caused by the sickle cells. My body is on the line, but I have accomplices, allies, and advocates on this journey. The two speakers before me are working to unravel the misconceptions and bring balanced and evidence-based messaging to discussions held in various venues. National, regional, and local organizations devoted to serving the needs of this community are actively working to bring about positive change. Though progress at times seems slow, it is steady. Together, we are lifting the voices of those who are unheard or too tired and weary to stand. We are speaking truth to power without ceasing. This sickled cell is a witness to ancestry of sacred blood that migrated over 85,000 miles, transported in enslaved bodies. They survived as I do. I am a sickle cell elder. Thank you.
Thank you so much, Ms. Jones, for your beautiful story. I'm greatly appreciated. I greatly appreciate all of these speakers tonight, that everyone has really brought different perspectives, and this has been fantastic. And Dr. Murray, had you posted something in the Q&A? Okay, so I guess there are a couple of questions here, and I will go ahead and read them. The first one is, how much have you found the who from the community or not to matter with regards to researchers administering and delivering the research? It's from Neil Hantry. I think that.
Yeah, thank you for the question. I just want to make sure I understand it. So is the question about, if the question is asking sort of who was involved in our partnership, it was, one of the things that we worked really hard to do is to make sure that we had a diverse group of representatives involved in our partnership. So some were, as I said, from the representatives of community-based organizations. One was the Philadelphia chapter of the National Black Leadership Initiative on Cancer. The other community group was the Southwest Action Coalition, which is a grassroots organization that operated in West and Southwest Philadelphia, sort of as like a, they were community organizers. And I think they're one of the reasons why they wanted to be part of our partnership was because they wanted to know what was going on at the academic institution at Penn. So, and I think that's a completely, valid and good reason for a group to be involved. So you know what's going on in your community, and you can have input and contribute to the process. So I think, in addition to like representatives from community-based organizations, we have residents who lived primarily in West and Southwest Philadelphia, because that's where our partnership started. But as our work grew and expanded, we sought out individuals, representatives from the other parts of Philadelphia. So, as I said, we really made the effort to have a diverse group of representatives from community-based organizations, and then individuals who were concerned citizens, who were concerned residents. So I think that ultimately made the work that we were doing to have greater receptivity and impact. And I think, one of the things that's really, we don't have a good measure for, we certainly measured impact in terms of changes in health behaviors. But what I'm interested in doing now is measuring community impact at a higher level of influence, right? So looking at individual behavior change is important, but I think we want to understand, through the work that we're doing, has in partnership with communities, has it really led to an improvement in sort of the social environment?
Yeah, so that's kind of half of the question that I was asking about others' involvement. I was also getting at the idea that, as you said, you were able to recruit like hundreds of African-American men to these projects. And do you think that, you being a person from the African-American community helped to push that kind of recruitment? And I guess I could ask the same question, you know, I guess of all the panelists in terms of like engagement with research, how important is it to have the people who are driving the research, you know, or does it really matter?
It's very important. I'm sorry, go ahead.
No, I agree. I mean, one concrete example is more recent. So with our team, I was the study investigator, but I don't think I was presented as like the face of the research team. Rather, our team consisted, as I said, of individuals who lived in West and Southwest Philadelphia and the whole city who worked as part of our team. And so, they were really the face of the study. I was just kind of like the data analyst person, working behind the scenes in some respects. But a more recent example is, for the work that I described at the very end, the basic behavioral science study. So there's a typical format, as many of you are aware, that you send an invitation letter that describes like the general purpose of the study, the procedures involved in participation. And as we use that method to recruit African-American breast cancer patients, we got minimal response. I mean, it was like, I was really concerned about our lack of accrual, and we weren't meeting our accrual metrics. So my study team, which actually happens to be all African-American women, made a suggestion that we should provide, a one-page flyer that has my picture on it, and then talks about, the study and how it's important to the work and the contributions that we ultimately hope to make in terms of improving breast cancer survivorship. That turned it around. I mean, that really made a big difference in our accrual rates. So I think it is important. And I think it, particularly if we're trying to recruit, pop groups that are very difficult to reach. Rick, did you have a statement?
Yeah, I was just going to piggyback on what you said. I firmly believe that that the research should the researchers should reflect the community in which they're researching, and it's important for many reasons. In particular, when you think about the African-American experience and the lack of trust and the distrust and the marginalization that the communities had historically with biomedical research, and then to think that white men are going to come in and ask questions and prod and take samples, and that's it's a very difficult task because of the relationship that's been historic, the historical relationship. And so when the community sees people who look like us, who have the same sensitivities, they share the same sensitivities, they share that that culture in the community, it does help. And it's helped me in my research. I've never really had any issues recruiting, because I would go into the community and I would say, look, I feel the same way you feel. There was a connection there, and it was an important connection, because for so many of them, they didn't feel any sort of value in biomedical research or seeing themselves in that research. And so how do we fix that? We fix it by bringing in more, we increase that pipeline, that diversity in the pipeline for biomedical workforce. We have to, or else we're going to continue to not be able to touch and reach all the communities in which we need to. One of the reasons why we have this lack of data on African Americans and Latinos and Native Americans is because nobody's focused on that. And those communities have, in many cases, withdrawn and not wanted to participate. A classic example is even the vaccinations. I mean, folks are waiting, watching and waiting, as opposed to feeling comfortable enough to say, look, I feel that our community, there's enough data here, we should get involved. So yeah, and representation, Neil, isn't just having a nurse come out, a Black nurse, okay, or a Black study coordinator. I'm talking about representation across the board, all the, you know, from top to bottom of these studies, whether it is all of us or some little study on you know, ingrown toenails, you know, coming out of the community college. I mean, it's you got to be representative of that community.
Yeah, I agree. So I also wanted to get Ms. Jones's input as someone who's been in research projects, how much of a difference that might make to you.
And the difference has been over time, the way that the outreach is done. And so like just sending a flyer or whatever, that's how it was in the beginning. And then it might have been, okay, we'll take you to dinner. We'll have a dinner for you all and present some information. But it seems to be most successful when there's an investment of time in whatever the community is, and not just for that period of the study. So you have to have an established relationship. You have to have, you know, a vested interest. You really have to want to know about me and what my daily life is about. I'm not just a petri dish. You know, I have a life outside of the petri dish. And so if I can see that you have some interest in my whole life, and I guess that goes back to all of the social determinants as well, it might be, I might be more willing, and I might be more willing to introduce others, because also too, you have to have a person in the community from the side that you want to investigate on, who's going to kind of maybe carry some water for you and to say, yeah, these people are to be trusted and what have you. So it takes some work. It's not, you know, just fly in and fly out. And that has changed over time. That attitude is changing over time. And I think that's why there may be more success.
Thank you. That's awesome answers.
Oh, thank you very much. And I guess the second question here is for you, Dr. Kittles. And that question was, thank you for your talk. Can you talk a little bit more about what you mean when you say that race and ancestry are often mediated by skin color? Is this in reference to disease-associated alleles on chromosome 15? What about other chromosomes?
No, maybe I spoke a little too fast with that. I didn't mean it that, if I said that, I didn't mean that. What I was trying to say was race is a predictor of several poor outcomes like poverty and poor health disparities. And in the United States, skin color has been used to create these racially stratified social groups that limit certain levels access to quality education, access, you know what I'm saying? So these socially stratified groups are really based on skin color. And even within the African-American population, we see this where skin color plays a role in terms of access and privilege. And so my point is that when we talk about race, we have to talk about racism, and racism is mediated through skin color. That's what I was trying to say.
Thank you. And then there's a question in research, how can researchers recruit minority individuals? I study autism and I'm very involved in the autism community. Regardless of my position, most of my participants end up being white males. What can I do to diversify and help minorities?
I think Ms. Jones's answer to the previous question really applies to that question as well. I think, you know, having a sincere desire for your research to have an impact in the particular community or group of patients, you know, is the critical starting point. And then, you know, working to develop partnerships that enable you to, you know, develop relationships that will be mutually beneficial. I think, oftentimes, researchers have approached community groups and have approached patients from the perspective of how can you help me? And I think, the goal for an academic community partnership, in contrast, is really about how can we help each other to address a really important and challenging question or issue. So I think Ms. Jones's response really, I feel like that everything she said applies here. Ms. Jones, did you want to?
No, I think, what you said and what I said, it's a two-way street. It's not one way, it's a two-way street. So it needs to be win-win for everybody concerned.
Yeah. And I think we really have one more question. We're running out of time here, but I thought I would ask this question with, how long do you think it will take the NIH to require researchers to incorporate diversity, specifically ancestry information, in their projects? They've already declared sex as a biological variable.
Yes, that's a great question. I think that's coming up really quickly. And it's something that I've seen change over time. When we first started looking at ancestry, there was a lot of hesitancy, a lot of pushback. But now people have to incorporate that in many of these genomic studies with African and Latino populations. And if you don't, then you're going to get dinged in, when your grant is reviewed or your paper reviewed. And it's just a matter of time before NIH really pushes that as a important ingredient to these grants.
Yes, and does anyone else, Dr. Hughes Halbert, do you have any guesses on that either? Ms. Jones?
Well, Rick is the expert in this area, so I agree with him. I think it's something that we're all, I think many researchers are cognizant of. I think as it becomes more and more a required element of research studies, I think it will naturally translate and be implemented into clinical settings.
Wonderful. Well, we really are at time, and I want to thank all three speakers for their tremendous job tonight. This has been really fascinating, and it would be so wonderful if we could keep talking for hours. I know that we have a lot of questions remaining, but this has been really fantastic. And I want to thank the attendees as well, and we're going to send you a survey. The surveys are really helpful for us in planning future events, so we ask that you all please complete those. And we also welcome you to join our email invitation list for future webinars. The recording of tonight's webinar, as well as the February 9th webinar, will be available in the next couple of weeks. And we will be sending out a note to you all when those are available. We hope that you'll join us on March 23rd, where the next topic is going to be on CRISPR technologies and the ethics and ethical discussion with Dr. Jason Haney and genetic counselor Sarah Huguenard. So thank you all very much tonight. These speakers, thank you. And to all of you all, have a good night, and we hope to see you again.
Thank you.
Take care.
Thank you. Bye-bye.
All right. Bye-bye. Thank you.